Now we understand what is enzymatic specificity. Specificity specificity refers to what? As I said, enzyme bind with the substrate. But how? The concept, two concepts are there. One is induced fit. Coastland is given. Another concept that is given by lock and key models. So the conformation, both enzyme and substrate, both are changing, then that will shows. So enzyme specificity refers to the ability of enzyme selectively bind, selectively bind and catalyze the specific substrate or a group of substrate. First sentence I am repeating, enzymatic specificity, if it is defined or explained or give meaning, so you can write like it refers to the ability of enzyme to selectively bind or catalyze a specific substrate or group of substrate. Another things, and the specificity of enzyme is crucial for the functioning of any biological system, as how it ensure that the correct reaction occur at right place and right time. Okay, so this is in applied in all the reaction what is happening here. I am talking with you, but what I am speaking at right place and right time. Similarly, here is also specificity is what is a crucial step for the functioning of whole biological system. It ensures that the correct reaction occur at right place and right time. Another same things. I am repeating this. Enzymes achieve specificity through a combination of structural features and chemical reactions changes. Okay. So one by one, if it is asked in the examination how they are asking, that I will give you. For example, in this slide, in my lecture notes, if they ask define the specificity of enzyme, their types, or describe their enzyme specificity types. So you can write like this: enzyme is a based on the ability of choosing a particular substrate. Ability of to choose a particular substrate and molecular recognition mechanism or regulation mechanism. This can attain only with the similar shape. This can attain or it achieve only with the similar shape between the substrate and the enzymes. As I shown in the previous lecture, enzyme substrate complex with the blue color. Sucrose is the example with the uh, hexagon or six-member ring structure. Similarly, which is the structural complementary. Enzyme specificity. Types of enzyme specificity can be categorized into seven groups. Here I have given only a six group. So one by one is bond specificity. As I described, three bonds are there: hydrogen bonding, complementary, then covalent bond. Then group specificity, which group is attached with that. Then substrate specificity, optical or stereo specificity, geometrical specificity, and cofactor specificity. So you can like write like this, and then explanation. What is bond specificity? How it is look like? So this type of specificity you can say or is said to be a relative specificity, or its specificity is less. Protein are specific to the substrate having a similar bond and structure. So only a bond and structure. For example, lipase. This lipase can hydrolyze ester bond between the glycerol and fatty acid in the fats. This is the basic example of bond specificity. Now group specificity. Another. Enzymes are specific to the type of bond. Remember, type of bond, and the group that surrounded it. These categories show more specificity than the bond specificity. This specificity shows more specificity, group specificity, bond than the bond specificity. The other name for these moderate specificity or structural specificity. In many literature, you will find moderate specificity or structural specificity. For example, you can find in the exopeptidases or endopeptidases in the protein. So substrate specificity, you know very well. So enzyme shows specificity toward the one substrate and one reaction in this type. For this type, specificity is very high. Or other name for, you can say, absolute specificity. In many groups, you will find absolute specificity. That I will teach you also in next lecture. Lactose, sucrose, and maltose have a specific function. Do not perform any other reactions. So, optical or stereospecificity, that is specific to substrate and their optical configuration. For example, L amino acid can only react on L amino acid, but not on D amino acid. This will specify the arrangement of atoms in the space, and specificity that is very said to be very high. In this case, you can say optical or stereospecificity. Now, geometric specificity. This has a less specificity as the enzyme can act as a different substrate. Remember, now enzyme can act on different substrate having a similar size or geometry. Example, alcohol dehydrogenase that can act on alcohol functional group like ethanol, methanol, and propanol. All three groups: ethane, methane, propanol, alcohol dehydrogenase. Now, certain specificity are cofactor specificity. So these has greater specificity. You can say that these act as a specifically toward specific substrate and a cofactor. Substrate with the cofactor. You have to remember this word. This reaction can be allowed only if the substrate and cofactor are the same and it is match one, and which is highly specific. This cofactor, if the cofactor is absent, for example, I am giving you, if cofactor is absent, enzyme remains inactive. It means they have a pair system. One is present, another is present, then the reaction is happening. If any single is absent, then the reaction cannot happen. So, the enzyme works according to their specificity and perform various biological and metabolic functions only if, even though many substrate molecules are present. So, this is all about the enzyme specificity and types of enzymes that can ask in the exam.